Okay, I'm going to get going. Good morning, everybody. I did remember today to bring my biochemical pathways, so I'm going to get some volunteers. You two guys are volunteering? Sure. I heard a lot of enthusiasm there. That and that. And you two guys can do the other part. Come on.
Oh, now I know what you're doing. See, this is why I'm a major, Daniel.
Move down a little bit. It says biochemical pathways. Okay, yeah, it's like the New York subway system or something. This is what's going on in your cells, okay? All of these are individual reactions going on in cells like ours. And some of these things, for example, I looked at it a minute ago. Down here you've got mostly, no, up here you've got mostly amino acid biosynthesis, and down here you've got a bunch of lipids being synthesized, and in the middle there is the process we'll talk about, glycolysis and the Krebs cycle. And unfortunately they've moved the second part of the process down to the bottom of here. What happens in mitochondria. There's a lot of molecular biology on this particular one, scheme 2. And the only point I want to make about this, other than you're totally responsible for all of this for the exam, that's cruel and inhuman punishment, is that every step on this chart, every metabolic reaction is catalyzed by an enzyme. So you should realize the importance of enzymes and why they get 2 lectures or so. So it's rather complicated, okay? And when you take biochemistry, unfortunately you do go into some detail about these things. So good luck when you take it. I won't make you responsible. The volunteers did a great job. Okay, so to summarize what I was talking about last time, very briefly, I was talking about enzymes, their structure, and what they do, how they work. The work they're doing is to lower the activation energy for a reaction to speed up the reaction, the catalyst. And as I said, unfortunately, they're very good about cleaning the desktop off, so my glucose is gone. Because reactions, although thermodynamically favorable, that is having a negative free energy change, they occur rather slowly. So enzymes are absolutely essential in living organisms to speed these reactions up. They do this by interacting with a substrate molecule, forming a complex at this special site within the enzyme, the active site, which is formed when the protein folds into its tertiary structure, or in the case somebody pointed out, for enzymes that have a quaternary structure, they would fold into their native conformation, and it would be in the quaternary structure of the protein. And at the active site, the bonds that are being formed or broken occur. There's a reaction, and one gets the enzyme back, so it can do it again, and the product formed. And at the end of the hour, I was talking about the induced fit mechanism, where both the enzyme and the substrate are altered slightly as they interact with each other. The enzyme, there's a good picture in the book of the enzyme hexokinase showing when glucose binds the hexokinase. You can see the structure, the cavity into which that glucose fits changes ever so slightly, but that's a significant change, making it easier for glucose to react with ATP in this particular case. Now, I like to spend a few minutes on something that is not often discussed in detail in books, and that is something which terms you will see in various textbooks. You will see the term cofactors and coenzymes. And I think these are terrible terms, and I refuse to use them. They don't say anything to me. They don't give you any idea about what's going on. Unfortunately, there is a compound we will talk about later, which is known as coenzyme A, so that's its name, so it has to be talked about. But what these compounds do, and the term I use, is prosthetic groups. Prostheses are things that help you, assist. I think prosthesis means assistance, assisting. So these are molecules which are attached to many proteins, not all proteins, carboxylases. But many proteins have either an inorganic, small, low molecular weight inorganic compound, or as you'll see in a minute, a transition metal ion, which aids in the reaction that this enzyme is carrying out. How many of you take vitamins? Fair number. Okay, do you know what's in your vitamins? Do you ever read the label? Vitamin, B1, vitamin A, vitamin D. They're all the B vitamins. There's C. Many of the vitamins, thiamine, vitamin B1, riboflavin, vitamin B2, niacin, B3, pyridoxine, B6, cobalamin, B12. Many of these compounds end up bound to enzymes in a modified form. For example, thiamine is converted to thiamine pyrophosphate. We'll talk about the enzyme, an enzyme that contains this compound. When I talk about respiration, we'll talk about FAD and FMN. We'll talk about these two. We won't really talk about pyridoxal phosphate. But these compounds are attached to proteins, and they are essential for the activity of the protein. They bind generally at the active site of the protein, which means they may most likely interact with the substrate of the protein. The example I've given you is shown in figure 84, which is for biotin. Again, you don't have to know the formulas of these compounds, but you should know what a prosthetic group does, because you will see that term again. So what's shown in figure 84 is the structure of the biotin molecule, and it also shows that to the left is the structure of the biotin molecule. To the right, it shows that biotin is covalently bound to a lysine residue of a protein. The proteins that bind biotin are known as carboxylases. Carboxylases transfer or release CO2 from substrates. And if you look carefully at the right-hand figure, you can see I've drawn a little box around one of those NH groups in one of the rings. That is actually where carbon dioxide, CO2, binds to the enzyme. So I think this is a very vivid demonstration of how a prosthetic group works or helps carry out the reaction. It's actually binding the substrate. It's, of course, at the active site if it's binding the substrate, but it is binding the substrate. And some of these other compounds as well, thiamine pyrophosphate is also in a direct binding of the substrate role. So prosthetic groups are important. If you remove a prosthetic group from a protein, is it going to be functional? No, it won't work. So they are absolutely essential. And you get a lot of your prosthetic groups from the vitamins that you take in the morning. These are all organic compounds. There is a larger group in fact, I haven't put them all on the board, of compounds which are called trace metals. They're present in living organisms at very, very low levels, but they're absolutely essential for metabolic activity because they are also prosthetic groups. But these are, we'll call them inorganic compounds. And I've put up here the sort of main ones. There are a lot more that are functioning in various systems. For example, iron, way back when I talked about hemoglobin, I mentioned when I discussed hemoglobin as a protein with quaternary structure, myoglobin a protein with quaternary structure, I said that that protein contains heme. And the structure of heme I gave you in this earlier lecture, and I mentioned specifically that if you look at the structure of heme, it's a bunch of nitrogen groups surrounding an iron atom. And the iron atom is the place where oxygen is bound as it is transported around cells. There's another group of proteins we will talk about extensively in respiration and photosynthesis, which are cytochromes. They contain heme, but in this case, the iron is undergoing oxidation reduction. Ferric, ferrous, ferric, ferrous. Copper, very common, found in various oxidases. These are oxidation reduction enzymes found in a protein important in photosynthesis that I'll talk about, plastocyanin. Zinc, carbonic anhydrase, an N. I mentioned in the last lecture, only because it has a bang-up activity, really good 600,000 molecules moved per second per protein. So I've got here the structure of carbonic anhydrase. If I can get the thingy shown up there. Great, okay. So this is carbonic anhydrase. It's a relatively low molecular weight protein, 30,000. And you can see in here, there's something going on there, something looks unusual. Zoom in. And what this shows you is, that's the zinc ion that's in carbonic anhydrase, and there are 3 groups around it. Those 3 groups are shown here. They're histidine residues in the protein. So this, there, okay. This is the active site of carbonic anhydrase. It has a zinc. There's a histidine here, histidine here, histidine here. And the mechanism of carbonic anhydrase, what carbonic anhydrase does is it takes CO2 plus water and it makes carbonic acid. It seems like a rather trivial reaction. So somebody said that reaction occurs in the absence of an enzyme. Yeah, well, it does. But the enzyme makes it go much faster. And what's shown in the middle of page 35, again, there's the structure of the active site, and it shows you to the right how the zinc atom is participating in the reaction. What it's doing is it's actually binding the CO2 and making the CO2 more accessible to OH, in this case, water, to form carbonic anhydrase, to form carbonic acid. In the case of an enzyme like carbonic anhydrase and some of these other proteins, the metal ion can be removed. It's rather easily removed from carbonic anhydrase. And of course, what you would expect when you remove zinc from carbonic anhydrase is the activity is going to decrease. In fact, it goes to 0. And then you can put the metal back in, and it rebinds, and the activity comes back. A very, very nice demonstration of the absolute essential nature of a prosthetic group. So the other 2 that I've just put up here, magnesium involved with enzymes that make or break phosphate groups, such as ATP, other phosphate groups. So that's a very central sort of trace metal. And finally, manganese, which is a more unusual metal, is found in various systems, but where it is truly important is in photosynthesis, where it is absolutely essential for making oxygen. Manganese is somehow involved in the specific step whereby water is oxidized to molecular oxygen, a reaction on which we are entirely dependent for our lifestyle on this planet. So that's why I talk about prosthetic groups, because I think they're very important. Now, I'm going to talk maybe for the rest of the time. We'll see. I'm going to talk about enzyme regulation. And there's an important point I want to make. Enzyme. Oh, I can't spell today. I'm sorry. the initial point I want to make, which is, one of the big points of this series of lectures, enzymes. All enzymes don't work all the time. And I was talking yesterday at office hours about this point. We all have the same DNA in all our cells, right? Yes. But we have many, many different kinds of cells. We have nerve cells, blood cells, liver cells, all kinds of cells, and they're all different. And I think they're different for two major reasons. One is that although the DNA is the same, the information content available to a cell, any cell in your body, is the same. In some cases, all the DNA is not used. You'll hear a lot about how DNA transcription, translation is regulated. Genes are shut off, genes are turned on. Obviously, that's going to affect what proteins are in the cell. But the second is that even though you may have all the same enzymes in some cells, A, B, and C, they're not all active at the same time. The activity of enzymes are regulated. They can be on, they can be off, they can be low activity, they can be high activity. So those two factors play out in the differentiation of cells that we have. There are a number of ways you can affect the activity of an enzyme. One of them, you're going to do this in lab next week, okay, is to, one, vary the substrate concentration. So when you are doing your amylase experiment, you're going to carry out this experiment where you measure the activity of the enzyme and you vary the starch concentration. That's the substrate for amylase. And what you see is a curve that you have seen before. It looks like that. Look familiar? Unless you slept through the transport lectures. Okay, this is a curve that at low concentration, there's a somewhat linear relationship between activity and concentration of starch. But then the rate plateaus off because the reaction saturates. Up here, saturation, I'll say. Because at some point, all of the enzymes in the sample or in the cell are functioning as quickly as they can, and adding more substrate does not increase the activity. Figure 87 shows that. The level at which the activity saturates is the maximum velocity. And then there's another component that is commonly measured when you measure enzyme kinetics, is what we're talking about. And that is something called the Km for the enzyme. 50% of the activity. The concentration of substrate, which gives 50% of the maximum activity. So you go along here and you come down to this point. That's a concentration of, let's say, 1 unit of starch. You would say amylase has a Km of 1 unit of starch. All enzymes obviously have a Km, but they're all different because all enzymes are different. When you see this kind of behavior, this is referred to as Michaelis-Menten kinetics, and it will be distinguished from a different kind of kinetics in a while. So what's the importance of the Km? Well, it gives you some measure of the affinity of the substrate for an enzyme. If you have an enzyme that requires 10 millimolar substrate for half activity, that's not a very, firstly, that's a burden in the cell possibly. You may not have 10 millimolar of the substrate around, so there's something funny there. But an enzyme that works at lower levels of substrate obviously require lower levels of substrate for maximum activity. So it's a relative number. It's not exactly a binding constant. It's not exactly what is called an affinity constant, but it is some measure of how readily a substrate binds to the particular enzyme that one is working with. Okay, but obviously if you vary the substrate concentration, you're going to vary the activity of this enzyme. If the substrate concentration drops to to a low value, you're not going to have very much activity. There are a couple other sort of environmental conditions, I guess I'd call them, where you can alter the activity of an enzyme. Again, you're going to do some of these in lab. Temperature, so 2 would be temperature. Most enzymes show this kind of behavior. Activity versus temperature. They go up and they go down. And you would expect for organisms whose basic physiology occurs where ours does, that would be the optimum temperature for our enzymes. But not all enzymes behave as we do. For example, there are enzymes in bacteria that can function basically in hot steam baths. If you ever go up to Yellowstone, Yellowstone's an amazing place. You've got all these bubbling, gurgling things that are going on with boiling water. And there are organisms who live in those baths. And the temperature is basically 100 degrees centigrade. They're boiling. And the enzymes are in there obviously can function. Our enzymes would not function at 100 degrees centigrade. Think about the behavior of this curve. Why does it go up? Why does it go down? I'm not going to tell you why, because it should be fairly obvious to you in terms of your understanding of protein structure. But it doesn't go up and stay up forever, in other words. You don't get a curve that goes up like this, and then it just stays up, and this is now 70 degrees, and this is roughly 40 degrees, and this is 20 degrees. It doesn't do this, and I want you to think about why it doesn't do that. Okay, I could take the same curve. Well, I'll redraw it. pH. Same kind of curve. Activity versus pH. This would be 7, roughly. So activity goes up to an optimal pH, an optimal activity at a pH that is around what you would expect physiological pH to be, and then it drops down again. Again, think about why this curve looks this way. It's a little different than the previous curve I had on the board. Not all enzymes have a pH optimum of pH 7. Enzymes in our stomach, for example, which experience something like 1 molar HCl, you would expect those enzymes to have a pH optimum down at pH 1 or 2. If you take them to pH 7, they won't work. So they're, depending on the enzyme. Okay, so these two factors come into play in regulating activity. The next thing that comes into play are inhibitors. And I like to talk about enzymes in terms of they work very well and they work very efficiently. They're almost magical. But there are compounds that can look like the substrate, competitive compounds, competitive inhibitors. Competitive inhibitors compete for the active site with the substrate. So they bind at the active site. And they prevent the substrate of the enzyme binding at the active site. So the activity will go down. Unfortunately, lots of compounds look alike. For example, you look at the amino acids. They all have an amino end, a carboxyl end, and then they've got their R group, but some of them look very similar. So it's conceivable that a competitive inhibitor for an enzyme involved in an amino acid pathway could be inhibited by a similarly structured amino acid. However, competitive inhibition is reversible if you add more substrate. Because if it's a competition between A and B for the binding site at the active site of the substrate, then usually you would expect the compound that is there is going to bind more effectively. So there is a reversibility to competitive inhibition if you can add more substrate to the system. That's what's shown in figure 8, figure 88. Figure 88 shows an enzyme. The inhibitor binds at the active site of the enzyme, no product formation. On the other hand, there's a second kind of inhibition, which is known as non-competitive. And the competitive or non-competitiveness refers to whether it's competing with the substrate. In this case, you've got an active site, but you have another inhibitor binding, another site on the enzyme that can bind an inhibitor. And when that inhibitor binds, the active site has to be altered, subtly, probably, not as big as is shown in the figure, but they're making a statement there. Obviously, the non-competitive inhibitor is affecting the activity, and the activity is driven by what's going on at the active site. So the active site cannot bind the substrate as effectively, even though the non-competitive inhibitor is not at the active site of the enzyme. Yes?
Experimentally, how would you differentiate between the two forms of
You would differentiate by the reversibility.
That's like the only test?
Yes. Or it's the simplest test. I was going to say this. This is not a process that you would expect to be reversed by the addition of more substrate. Okay? Okay, now we come to a subject that sometimes causes a lot of pain. There are some enzymes, I'm erasing the wrong figure as I usually do. There are some enzymes that when you measure the substrate versus activity profile for these enzymes, they show a behavior not like this, but they show a behavior like this. This is referred to as sigmoid kinetics. It doesn't have the name of the people who studied it. Michaelis and Menton were British scientists in the 1930s who were enzyme kineticists. That's why they got their name associated with the kinetic trace. So you get this S-shaped curve. The explanation for this sigmoidicity is cooperativity, which I'll expand that. Cooperative binding of substrate. And if you look at figure 90, that is what is being shown in figure 90. You have a protein that has quaternary structure there, right? 4 subunits. And you can see that the subunits, they have an active site. Each of them has an active site. When you bind low concentrations of substrate, there's not very much activity. But the enzyme changes conformation to make subsequent binding of substrate easier. So the first few substrate molecules bind and the enzyme starts to change shape. It makes the active sites more available, and this binding of substrate and activity go up in this particular way. Now you'll only see this in an enzyme that has quaternary structure, that has more than one active site, because the active sites are interacting with each other. So this is a very common thing that one sees in biological systems with enzymes. And there's an extension of this, which is known as allosteric interactions. I'm going to modify this curve. I'm going to get rid of this. It's possible to shift this curve like this or like this by adding allosteric activators or allosteric inhibitors. And there's a rather complicated scheme that's shown in figure 91. Trying to illustrate this, you have this enzyme in two forms, a highly active form and a highly inactive form, and an activator will shift this equilibrium to the more active form, and the inhibitors will shift the enzyme into a less active form. So there's a rather complicated interaction going on here, but the allosteric inhibitors and activators are not binding at the active site of the enzyme. So what are they? They're non-competitive inhibitors, right? What are they actually doing to the activity? Well, I've drawn these figures to make it easier, I think, to understand. You have maximum activity up here. So this is 100%. So the Vmax for these three forms of the enzyme are the same. But what's changed? What's changed is the Km. So you've gone from a Km of this to an activated system to that. So this enzyme you would expect to be more active because it is certainly requiring less substrate to have 50% of its maximum activity, whereas this form of the enzyme is going to require, let's make this 1, 10, and 50, is going to require 5 times more substrate to attain the same 50% of its maximum activity. So these shifts can occur depending on the presence of substrate plus allosteric effectors, they're called. There are also some enzymes, and this is explained at the bottom of this figure, and it's not all enzymes, but some enzymes actually have two different kinds of subunits. I've tried to draw my own pictures. They have squares, and then they have circles. Oh, I only want 4 circles. 1, 2, 3, 4. Okay? So we've got 2 different kinds of enzymes, 2 different kinds of protein subunits in this enzyme. These subunits are called catalytic subunits. They bind the substrate. They don't bind the inhibitor or activator. There are regulatory subunits in these proteins which specifically bind the allosteric effectors. So it's a more complicated system than, say, if I wanted to have an allosteric regulation of an enzyme like the one shown in figure 90. And again, there are these very subtle shifts in the shape of the protein. Why do you get this behavior? You get this behavior because these compounds are altering the conformation of the protein. And there's subtle changes in structure. The protein is unfolding. I don't want you to get the idea the protein is unfolding completely and magic is happening. There's subtle changes, but the binding of these compounds can alter the active site of the protein so that the activity is either goes up or down. The classic example of allosteric behavior are in these pathways like amino acid biosynthesis pathways in the figure that I had, where you have a series of reactions where you start off with one compound, A, and it's converted to B, to C, to D, and this is the final amino acid, say. Regulatory and catalytic I'll write that down. Regulatory, those bind the allosteric effectors because there are activators and inhibitors. And C are catalytic. They bind the substrate. Okay? So this is the final, let's say it's an amino acid product. In these pathways, it's very common for this final product to inhibit this first enzyme through an allosteric inhibition pattern. And what happens in these pathways is you have amino acid being synthesized, the product level goes up. When it reaches a certain level, it inhibits this enzyme and the pathway stops because it says, I've got enough amino acid of product. I don't need it anymore. So it shuts down the whole process. And of course, you don't form any B, C, D, or E. Then the amount of this is going to drop because the pathway is shut off. And it's going to start up again because now this enzyme, the amount of inhibitor required to inhibit is dropped enough so that enzyme A can start up. So it's this loop, feedback inhibition, which is very common in many pathways, multi-enzyme pathways. It's a very, very neat sort of way of regulating the activities of these enzymes. Oh, let me come back to inhibitors. The classic inhibitor system is with the enzyme that is the terminal enzyme in respiration. We'll talk about this in much more detail. This is an enzyme that reacts with oxygen. During respiration, there's one place where oxygen comes into play, reacts with an enzyme called cytochrome oxidase. So you have O2 as basically a substrate. Unfortunately, there are a number of compounds like carbon monoxide and hydrogen cyanide, okay, that look to this enzyme to be like oxygen, believe it or not. Maybe carbon monoxide may be surprising, not less surprising, but cyanide. This is why cyanide poisons us, because cyanide comes in and it binds to this terminal enzyme in respiration, and unless one can get more oxygen into your system, it's a very effective inhibitor of respiration, and it's a competitive inhibitor with oxygen. So there are compounds that are very dangerous, and if you encounter them, they often will kill you, period. So this is kind of the classic example of an inhibition, two compounds that are may not look chemically the same, but electronically there is some similarity between oxygen and carbon monoxide, or HCN. Carbon monoxide has the same terrible effect, binding at the same place as the oxygen binds. Okay, I've only got 10 minutes, but I'm going to start on the next subject because it's interesting and it's important. There is a whole nother mechanism, series of reactions, processes involved in the regulation of enzyme activity. And they involve covalent modification of proteins. What I've talked about so far have not been covalent modification of proteins. They've been interactions where things bind rather loosely to proteins, and then after some activity, some occurrence happens, the inhibitor might leave, or it might not leave so easily. But now we're talking about covalent processes. Figure 92. Figure 92 off to the left is not terribly important, but it shows an enzyme, a protein being made, translated. That's a ribosome, believe it or not. And that protein is released, and then there is now something called post-translational processing. This is not the same as post-translational import. processing. And okay? And the first thing that it shows at the top of that figure is, no, the middle of the figure. It shows a protein and it shows glycosylation. Well, glycosylation is a covalent modification of a protein. So glycosylation is the addition of sugars. And we've talked about that. You all know about this process. In this case, this is a process that is involved in targeting proteins to their respective place. So I'm not going to talk about it in relation to enzyme activity and enzyme function and enzyme regulation, but it is a post-translational process. Addition of sugars is important for targeting, it says. The top one I will talk about, and then I'll probably stop. Proteolysis. There are a number of enzymes, and they tend to be enzymes involved. They're proteases. They break down proteins, and they're involved in digestion primarily. And they go by the names of trypsin, chymotrypsin, okay, those are two of them, where the enzyme is made in an inactive form. The inactive form is known as chymotrypsinogen. This is active. And this is chymotrypsinogen. This is an inactive form of the enzyme. That's the way it's synthesized. Trypsin. Trypsinogen. These are inactive forms of these enzymes. And at some point in the life of these enzymes, there is a proteolytic processing. That means part of the enzyme is clipped off. What is shown in figure 93 is the 245 amino acids in chymotrypsinogen. It's inactive. And then amino acids 1 to 15 are removed, and this is converted into the active form. Similarly, this happens with trypsin. And as I said, it's a covalent process. Once you remove these amino acids, you can't put them back. It's turn it on. It's a turn it on process. You don't turn it off again. There's a third process, series of reactions, which I'll just mention, and then, so this is one, and then I'll come back to it on Friday, and that is protein phosphorylation. And protein phosphorylation is very common, and it has the advantage over proteolysis that it's reversible. The term that is used for the proteins when they're in their inactive form, this is an old term, but you still see it. These are called zymogens. So you will see, if you see the term zymogen, you should turn a little light on and think that's the inactive form of these proteases that are involved in the digestive process. Okay? Okay, I'm going to stop because protein phosphorylation is so important. It's better to talk about it at the beginning of the hour than the end of the hour.